Alright, this is Big Picture Anatomy with a look at the cellular and molecular structure of a skeletal muscle cell and how it is that the filamentous proteins inside the skeletal muscle lead to a muscle contraction. First, let's take a closer look at the structure of the muscle cell itself and how the contraction is initiated. Remember that the skeletal muscle itself is very large compared to the tiny, barely visible in a light microscope motor neuron axon that brings an action potential down the axon to the skeletal muscle, where it synapses with the skeletal muscle at the motor end plate, or the neuromuscular junction, or synapse. What happens when that action potential arrives is that, looking a little bit more closely, right at the neuromuscular junction, acetylcholine is released, and the release of that acetylcholine, which we abbreviate ACH, causes an action potential in the muscle cell membrane. So if we zoom back out, what we'll see is in the muscle cell membrane, now you have an action potential being propagated or moving every direction along the entire muscle cell membrane, or remember what we call the sarcolemma. That action potential also dives down into the muscle cell through these extensions of the muscle cell membrane called the T-tubules. There's a couple examples of T-tubules here. When the action potential arrives at the end of the T-tubule, there's a concentration around the T-tubule of the sarcoplasmic reticulum, that internal endoplasmic reticulum equivalent in muscle cell, but with the special role of sequestering calcium. When the action potential arrives at the sarcoplasmic reticulum, the calcium that has been sequestered is released out into the rest of the muscle cell. Now, most of the muscle cell, everything else, is filled with these filamentous proteins. The filamentous proteins themselves are actually actin and myosin. That's the name of the two proteins, actin and myosin. And when the calcium is released from the sarcoplasmic reticulum, it triggers the filamentous proteins to slide against each other, in causing that sliding model of how muscle cells contract, and that's what we want to take a slightly closer look at. So if we zoom in on the filamentous proteins and blow up one of these now molecular structures to very high magnification, what would really be something you can't very well see in a light microscope, this is what we would see. So this is now a molecular level structure called a sarcomere. Again, the details of this you wouldn't see in a microscope, but the biochemists tell us this is what these proteins look at. And if we look at them a little bit more closely, what we're going to see is that we have a very highly organized structure to the filamentous proteins and the way they fit in with each other. The myosin protein, we call the thick filament because it's thicker. The molecular structure that's thicker is surrounded by a lattice work of actin proteins, which we sometimes call the thin filament because, of course, they're thinner. Together, that actin-myosin construct that allows for muscle contraction is called a sarcomere. An actin-myosin unit is called a sarcomere. Now, what happens in the sarcomere is that in the presence of calcium, the myosin heads form cross bridges or links over to the actin on either side and at either end of the myosin protein. And there's a little bit more complex three-dimensional structure to this, but this is how we can represent it in 2 dimensions. And those cross bridges link over to the actin, and I like to think of them as rowers, like in one of those Viking boats, and they pull that actin in, but they pull in from either side, and they literally physically row as little molecular structures in the presence of calcium. They row the actin in from either side. And what happens is then we gradually see that the actin is pulled in. You get more and more rowers grabbing onto the actin as it comes in closer contact with the myosin filament down the middle. And then eventually what we see is that the entire sarcomere, or the entire actin-myosin unit, gets pulled in about 20%. And if you then take those sarcomeres, or actin myosin units, and put them back into the overall structure of the muscle itself, what you'll see, or what we have, is then what are literally millions of these sarcomere units, all lined up inside the muscle cell and also linked end to end all along the length of the muscle cell. And they all act in unison in the presence of the calcium that was triggered to be released by the action potential in the muscle cell, all pulling in or contracting, leading to the entire muscle cell to pull in or contract about 20%. And that is our sliding filament model for muscle contraction. So, just to summarize, these are the steps in muscle contraction. We have an action potential coming down the motor neuron. That's the first step. At the neuromuscular junction, that action potential is transmitted to the sarcolemma, or the muscle cell membrane, and is transmitted throughout the muscle and into the muscle through the T-tubules. That in turn triggers the release of calcium, which has been sequestered in the sarcoplasmic reticulum, and the presence of the calcium in the muscle cell around the actin and myosin filamentous proteins, or the sarcomeres, causes the myosin proteins to form cross bridges with the actin that work at a molecular level to pull in or row in the actin from either side, causing therefore a shortening in the overall length of the sarcomere. And when you put all those sarcomeres together back in the muscle cell working together, a shortening of the entire muscle. That then is the cellular and molecular structure of a skeletal muscle and how it works in the sliding filament model for muscle contraction from big picture anatomy.